right, good morning, everybody, and welcome to the 2022 version of IIDR rounds. I'm Laurie Burrows from the Institute for Infectious Disease Research. Our speakers today are Zain Chagla and Jake Magolan. I believe Jake's speaking first. So Jake is an associate professor of biochemistry and biomedical sciences at McMaster. His lab specializes in hit-to-lead medicinal chemistry, which is a key part of the complex and interdisciplinary process of the discovery and development of new medicines. And today, Jake will be speaking to us about the discovery and development of COVID-19 therapeutics. And Zain is an associate professor of medicine at McMaster University. He is an MD specializing in internal medicine, tropical medicine, and infectious diseases, and over the last 2 years has been a leading voice in public education about COVID-19. Zain will be speaking to us today about the early experience with COVID-19 therapeutics and barriers to their large-scale implementation. So I'm very excited to hear your talks today, gentlemen, because I was one of the unfortunate Christmas Omicron recipients. I have recovered, but the therapeutics were certainly on my mind, so I look forward to your talks. All right, Jake, I will hand it over to you.
Thank you very much, Laurie, and thank you to everybody attending for waking up early. Can you confirm, Laurie, that you guys can see me and my screen and everything is okay?
Yes, looks good.
Okay, so we're going to start with a quiz, which, because I don't see the faces of anybody watching, will be perhaps a little bit awkward. But just for yourselves, here's a little multiple choice question for everybody. Which of these molecules are the following molecules? One of them is called Repurposed, which is Pfizer's new antiviral that inhibits a viral protease. The second, or one of them, is called Mullen Ipravir, which is Merck's new antiviral pill. Both of them kind of FDA approved as Christmas presents for the world in December. And this molecule induces RNA mutagenesis by essentially confusing one of the enzymes, one of the viral enzymes, the RNA-dependent RNA polymerase enzyme, and causes it to make mistakes. And then the third molecule is hydroxyquinoline, which is just an old malarial drug. Okay, so it is my hope that everyone or most of you guys can identify these three compounds. And if not, then I'll help. Generally speaking, a protease will kind of look like a peptide, okay? A polypeptide. This is the protease. It mimics the substrate of a protease enzyme. Generally speaking, an RNA polymerase enzyme substrate will look a little bit like RNA, and this has that nucleotide look to it. Of course, it's a little bit different. The base is modified here, most notably. And then the hydroxyquinoline, the quinoline that comes from that heterocycle, which is called the quinoline, okay? So there you go. So a little bit about these things. Molnupiravir, based on one of the clinical assay papers that I read, reduces hospitalization about 50%. It was authorized in the UK in November, in December by the FDA, but not unanimously, a 13-10 vote. Perhaps Dr. Chagla will discuss a little bit about why that is the case. And Canada is a little bit behind. One treatment course in this case is 8 grams of this substance. So Merck needs to manufacture a lot of this molecule in the coming year if it's going to be used globally. Hydroxyquinoline was proven useless against COVID infection after more than 190 clinical trials, which is an absurd number of clinical trials. And I'm going to talk a little bit more about that. Certainly far more than was required to prove that that was not effective. And this protease from Pfizer is really a remarkable drug, custom built for this particular infection, and it reduces hospitalization by 88% when administered within 5 days of symptoms. And it's now authorized in Europe and the US and Israel and the UK, but still not quite in Canada, but I imagine it will be in Canada very soon. In this case, a single treatment course is 3 grams, and Pfizer has stated that they will manufacture 120 million courses, which is 360 metric tons of this molecule in 2022, which is a lot for a molecule of that complexity. It's a kind of a Herculean process, chemistry and manufacturing task that they are doing right now all over the world, including at contract synthesis companies in Canada that I'm aware of. Everything is happening all at once everywhere to manufacture large amounts of this molecule. As far as the entire medicine cabinet as it stands today, those 2 drugs are the most recent, and they're the sort of orally bioavailable pills. There are 5 different companies that have produced monoclonal antibodies, all of which have to be administered by infusion. And then there are some repurposed drugs that have been approved for use. The first of them, Dexamethasone, is a corticosteroid that actually is not formally approved, but is being widely used off-label ever since a clinical study in England in June of 2020. And then after that, these JAK inhibitors and IL-6 antibodies that are typically arthritis drugs that are immunosuppressive have also been proven effective. And then 2 others, remdesivir and convalescent plasma, that are approved in the United States, and remdesivir is also approved in Canada. I put red boxes around them because there's warnings against their use from the WHO and in England because they don't work. In the case of convalescent plasma, sort of the clinical evidence appears to show that it doesn't work at all. And in the case of remdesivir, it doesn't work for hospitalized patients. Okay. Here's a timeline, and a lot of this talk is going to show this timeline with these 24 months across the middle, January to December of 2020 in blue, and January to December of 2021 in gray. And this is a little bit of a history lesson for everybody watching, and it's recent history, and it'll feel familiar. And it's tragic history, of course, because of the graph. These are the Ontario deaths, COVID deaths. We reached peaks of 7-day average of 60 deaths in Ontario per day in April, May of 2020, and then again in January of last year. And since then, it's been considerably better. But again, we're in a spike now. So, the jury is out about whether that number will start to rise in the coming weeks. This all started with a new virus identified on January 7th, the first death reported on January 11th, the first case in Canada on January 27th. And you guys recall McMaster closing its residences and stopping in-person classes on March 13th. I believe I attended a McMaster volleyball game on March 7th, and that's the last public activity that I recall doing in a large crowd since then. So I put this calendar up there because I want to emphasize how remarkably fast some of this development has occurred. There's no precedent for it in medical history, this pace of brand new medicines and vaccines. So in the case of vaccines, Russia released a vaccine in August, which is just a remarkable story. And I don't know anything about that story, but I imagine that there's a story to tell there, and it will be told in the coming years. And then the rest of the world started approving brand new vaccines in December, starting with Pfizer, Moderna, and then AstraZeneca and Johnson & Johnson in the US and Canada. As far as therapeutics, I'll put them on the bottom. So about 10 months after this new virus was identified, Donald Trump received a new monoclonal antibody therapy that targeted the spike protein of this virus, the specific spike protein. And that's really quick for something like that to be available for compassionate use, as it was in his case. The next month, the FDA authorized it for emergency use, and it was the Regeneron combination of 2 monoclonal antibodies. And if I was to show you all of the new medicines that have been approved on the bottom of the screen, that monoclonal antibody was the first. And after that, 3 others. The Lilly one was approved first as a single monoclonal antibody, the bamlanivimab. Wow. And then it was approved again in combination with another one that Lilly acquired from another company. And these have been steadily coming out every couple of months, new approvals, and they've saved a lot of lives. And in December, of course, the orally bioavailable pills were approved, and now these are being manufactured en masse. Notably, the monoclonals target the spike protein, which is different, considerably different on the Omicron variant than the previous variants. The Regeneron and Lilly antibodies don't work very well for the Omicron variant, but the GSK AstraZeneca ones appear to retain their activity. In addition to those brand new medicines, dexamethasone has been used since about June 2020 in hospitals. Remdesivir was approved, first authorized in the States in May and then approved in October. Convalescent plasma was approved in the United States in August. And then these immunosuppressive, the small molecule drug and the antibody. And I imagine all of them are used clinically in the US. I don't know whether or not they are used in Canada. Just so I don't make all of this completely American, these are the drugs that are listed on Health Canada as approved presently. Remdesivir as of July, and then 3 of the monoclonal antibodies, the single antibody treatment from Lilly, not their combination. The UK has approved 2 monoclonals and has already approved both of the small molecule pills. We haven't done that yet, but I'm certain that's coming. All right. So if I go back to the US for a second, I want to highlight one other thing that was, I think, somewhat of a shameful moment in the history of the FDA. They authorized, under political pressure, hydroxychloroquine for emergency use in COVID after, the president of the United States, even prior to authorization, was lying and saying that it was approved, and prior to any clinical evidence of its use, of its effectiveness. And then that authorization was quietly revoked on June 15th, and it's hard to even find that on the FDA website now. Okay, so I want to address how this happened so quickly, and I think a really important story that I think others will be able to tell much better than me, but I'll try to do it quickly, was the story of this genome sequence. It's an interesting story, and I read about it in preparing for this talk, and I thought I would share it with you. So on January 11th, the sequence of this was posted online from a patient admitted in Wuhan on December 26th. And there was a story published in Time magazine in August of that year about this professor, Professor Zhang, at this research institute in Wuhan. No, I'm sorry, he wasn't working in Wuhan. The sample was shipped to him at his research institute. On January 3rd, he received the sample. On January 15th, the sequencing was done. There's a quote in this article about him. You can imagine, perhaps you can put yourself in the shoes of somebody. In hindsight, this looks really obvious, but at the time, it was not so obvious that this is urgent and needs to be urgently released. And the quote says, he had 2 suggestions: that we should take some emergency public measures to protect against this disease, and also that clinics should develop antivirals. And then on January 11th, at an airport, he was called by a professor in Sydney who asked permission to release the genome publicly. I asked Eddie to give me 1 minute, and then I said, okay. And then for the next 2 hours, he was flying while this was released on virological.org. Okay. There was a little bit of drama about the Chinese government being surprised by this and closing down his lab for a week while they confirmed his protocols to make sure everything was authentic. But the bigger criticism after the fact was the 6 days between when his lab first sequenced it and when it was published. And in fact, it's now clear, and this article made it clear, that on January 5th, the genome was actually uploaded to the US National Center for Biotechnology. Professor Zhang is quoted in this article saying, "We posted it on January 5th, and the United States certainly knew about it at that time." But the NCBI takes a few days to look at these submissions. So the decision to also publish it online independently really facilitated things. And I'll explain why that facilitated things. There were a lot of, this is my structural biology appreciation slide. I don't know Sarah's here, but there were a lot of researchers that were already experts on structural biology of coronaviruses that were desperately waiting for a genome sequence to become public. And when they received this genome sequences, they got to work on expressing these proteins, modifying them so that they can be suitable for cryo-EM crystallography. And on February 17th, the spike protein structure came out, and soon afterwards in March, the main protease structure, and then the RNA polymerase structure. And these are all drug targets now. And as soon as the structures were available, they were all immediately used for computational sort of drug discovery and optimization in medchem departments around the world. So the pace really is something special here. It's almost surreal that this was possible this quickly. The prettiest picture that I have found of this sort of proteome is from a professor at Michigan who does in silico protein folding, and his in silico folding created all of these images. But the virus is a single strand of RNA with 30,000 bases. It codes for 29 proteins. And there are a few that automatically stood out as drug discovery targets. And the three that I've circled are sort of foremost among them. But I want to explain why they stood out, because I think it's important to acknowledge that the main reason why these vaccines and these drugs were discovered within 1 year and came to market within 1 year is all of the research that was done since 2003 with the emergence of SARS and MERS. And there's a great article from ACS Central Science that sort of combines all the research. It came out in March of 2020, all the research on previous viruses, on these two previous viruses that may be relevant to COVID-19. So just for those that are not familiar, the origin of, I'm going to draw with a pen on this, see if this works. Yeah, okay, so the SARS outbreak was in 2002. It caused 700 deaths, and it was in Hong Kong, China. The MERS outbreak was in 2012. It caused 900 deaths, and that was linked to Saudi Arabia, right? And then, of course, there's some common cold coronaviruses, and we are in the sort of genetic space relatively close between MERS and SARS, and they're both beta coronaviruses. And there was already a really good understanding of what these viruses did and how they behaved and how they were structured. And in this ACS Central Science paper in 2020, Cynthia Liu and her colleagues show that there were already 500 patents just on biologics, vaccines, antibodies, and cytokines prior to the emergence of this virus. One of these was this patent from Moderna, describing an mRNA formulated in cationic lipid nanoparticles that already worked in white rabbits immunized against the MERS coronavirus. By February 24th, Moderna had the first batch of this vaccine ready to go and shipped to the NIAID. And all of that was possible because the work was already ongoing because of these previous outbreaks. In addition to that, the key proteins and their roles were really well understood. So these are the key proteins. There are two proteases. There's the polymerase, there's the spike protein, and then there's a couple of others that are sort of known to be drug targets, and they were already known, and that was very well established. As a matter of fact, the number of patents targeting each of these proteins at the time that this paper was published in March of 2020 was already pretty large, and the number of potential drug candidates that appeared in these patents was also, on the order of multiple thousand. And by far the main drug target for these previous two infections was the main protease, or the 3CL-Pro. I'll briefly skip over this, but I just want to emphasize the massive amount of previous work. These are all patents relevant to these previous drugs that were identified as potentially useful for COVID-19. And there's a lot of new small molecules that were already known that were targeting, the polymerase and the proteases and so on. So that is the sort of the best explanation, the rationale for why this was able to move so quickly. I think we're fortunate in a way that those previous outbreaks spurred a lot of research and innovation. And then, it could have been better, of course, but it also could have been a lot worse. So I'll talk briefly about the drugs. First of all, the monoclonal antibodies is outside of my area of expertise. So I'll point you, if you're interested in their mechanism of action and their method of synthesis and discovery and optimization, to a really good review in Nature Reviews Immunology that was published this year. And then I'm going to go on to this drug. Okay, so this is a combination of the newly discovered protease inhibitor with an old drug called ritonavir. And ritonavir, it's called a metabolic enhancer. It essentially inhibits a cytochrome P450 enzyme, the 3A4, and that enzyme is responsible for sort of the initial breakdown of this drug and many other proteases, including some drugs that are used in HIV. That's the way this was initially discovered. Actually, ritonavir was initially developed as a protease inhibitor. Just coincidentally, it was found to inhibit the metabolism enzyme that tends to metabolize protease inhibitors. And so now it's used with a lot of drugs as an additive to sort of extend the half-life. So it was almost always a given that when a new protease inhibitor came to market, it was likely going to be in combination with ritonavir. There are two papers that were published that are most relevant to the discovery of this molecule. The first was published in the Journal of Medicinal Chemistry in 2020, and it's about a precursor to this compound. It's called the discovery of a ketone-based covalent inhibitor. And this compound is old. And I think Pfizer had been sitting on it for maybe a decade without publishing it. And then they sort of dusted off all of that research and wrote it up and published it really quickly in October 2015. This was their top protease inhibitor for SARS-CoV-1, which was that 2002 SARS outbreak in China. And it wasn't a great drug. And the main problem was it was not orally bioavailable. It had to be injected, and it had poor permeability. So it's actually injected as a prodrug to help with cell permeability. But its discovery was a result of a lot of structure-based optimization around the active site of this protease. And this paper describes a lot of that medchem. And it was really quite a bit. They made a lot of molecules to get to there. And I'm not going to go into that today. But importantly, they had a good starting point. And that compound, the injectable prodrug, where this alcohol is covered up by a Laval group, is actually in clinical trials right now. But then they went to work on optimizing this molecule, and the result of that is the one that's now FDA authorized. And that work was published on November 2nd, and published in its issue as a Christmas present, kind of at the same time as the drug was released. And so that paper, I'll just highlight one slide. I'm sort of short on time, but I'll highlight one slide from this paper, which is, they started with this original lead and its potency against this protein, and the FRET assay that they have, was really good. It was sub-nanomolar. This is the efficacy at preventing cell death, let's say, cytopathicity in viral cells. That was also sub-micromolar. This is considered really good. This is a measure of permeability, which was bad. This is clearance in human microsomes. That was totally fine. and also clearance in rat. Sorry, this is not in a microsome assay, but in an actual rat in vivo clearance. That you want this number to be below 25. You want this number to be about below 50, so they're both fine. But the oral bioavailability was awful, and that has a lot to do with the permeability. These are often related. Okay, and they went to work on this molecule not to optimize efficacy, but to optimize these key PK properties. And they made changes to the molecules that I've shown in yellow here. Some pretty substantial changes. Frankly, they kept very little of the original molecule, and the result was a loss in biochemical potency, but an enhancement in vivo potency in cells. The permeability improved. The half-life actually got worse, right? So this is worse. More of this drug is metabolized, and that's another reason why it's administered with that metabolic enhancer. The rat clearance is good, but most importantly, the oral bioavailability is actually really good. So that was enough. This is a much improved drug. I'm not going to show you the synthesis as much as I would love to. I'm just going to break it down like this in the interest of time. It's made from essentially three amino acids and one cap, this trifluoroacetate cap. Two of these amino acids are no joke. This one here and this one especially. Pfizer will have to develop bulk scale syntheses or contract out enormous scale syntheses of those, and that will not be cheap. Overall, we're talking about maybe a 10 to 12 step synthesis with protections and deprotections. And if you want to make 100 tons of a molecule like this, it's a lot of work and it's an expensive task. It's not a simple compound, and the dosing is really high for this molecule. It's dosed like like an antibiotic or ibuprofen. It's not dosed like a kinase inhibitor or an antidepressant, which are typically much lower dosing. All right, the last thing I want to cover is this issue of repurposed drugs. I'm going to start with this public service announcement. A paper came out in February 4th, and that paper was in Cell Research, which is a high impact journal. It was a perfectly reasonable paper. The title was "The Remdesivir and Chloroquine Effectively Inhibit the Recently Emerged Novel Coronavirus." And if you look closely, it says that the EC90 value of chloroquine is almost 7 micromolar, which makes it a pretty bad drug. They say it can be clinically achievable, as demonstrated in the plasma of rheumatoid arthritis patients who received 500 mg. In any case, this paper was one of many sort of early papers that were not even animal studies that got people excited about this particular drug. I like this Nature Human Behavior paper. Reading work like this makes me think that perhaps I should have gone into human behavior research. I'm fascinated by it. But this is another public service announcement. And I don't know what the solution to this problem is, but I think it's something we should all be thinking about. The nature of this emergency warrants accelerated publishing, but measures are required to safeguard the integrity of scientific evidence and to sort of bridge the gap between the general public enthusiasm about science that they're excited about and the quality of that science, or the sort of the rigor of it. There were a lot of molecules that were predicted to potentially work. And a best case scenario, of course, would be that an FDA approved drug that already exists and is available can treat this virus. And all of them that have any basis for that hypothesis deserve to be tested. And they were. 644 drugs were trialed in 1,300 clinical trials. And this is as of February 2021. This is from the FDA website, this PowerPoint presentation I found. But too many of them were on the same compounds over and over again. So hydroxychloroquine is the worst offender in this category. 190 trials, 71 on azithromycin, 50 on ivermectin. And the ones in yellow are the ones that ended up being approved and working, right? Or at least being approved. Dexamethasone, was tried in this way and then used and saved lives, and so was tocilizumab. The UK government has on their website a list of ineffective treatments. If you're interested in looking at some of these for yourself and seeing what this UK government considers evidence that this is ineffective, it has this information and links for a lot of these things, including convalescent plasma. The New England Journal of Medicine in 2021, remdesivir, hydroxyquinone, lopinavir, interferon regimens have little or no effect on hospitalized patients. Okay, when something does work, like dexamethasone, the results look like this. So 160 people per 100 died, and then with methazone treatment, it was down to 126 per 1,000. I'm sorry, and that's considered efficacious, right? I like that the WHO puts this certainty of evidence, quality of evidence tab in here. And they did the same thing for these IL-6 inhibitors. In this case, the certainty of evidence is high, and both of these are approved. The World Health Organization strongly recommends corticosteroids and these IL-6 inhibitors, but has recommendations against steroids with non-severe COVID, certainly against remdesivir, against hydroxyquinone, against lopinavir, and against ivermectin, except in the context of a clinical trial. This stuff is out there now. I don't have time to talk about this. Is a really interesting paper that just came out over the holidays by our own Professor Ed Mills, who was involved in some of this clinical trial work of repurposed drugs. And this is about fluvoxamine, which is an antidepressant, which appears efficacious in this recent trial. So I wonder if that'll be added to the list in some places. It's really inexpensive medicine. I'm going to finish by acknowledging the people on this slide and apologizing to you guys that I just sort of decided not to talk about any of our ongoing work. So our lab at McMaster, in collaboration with Professor Wright's lab, Matt Miller's lab, and Mike Tyers and Anne Marnier at Montreal, are doing some medchem and discovery work in this space. But in this short talk, in this format, I decided to sort of focus on the published, more clinically relevant research. But I do appreciate all the work that all these folks have put in, and we'll certainly present it and publish it in the future. And with that, I'll stop, and thank you for your attention.
Thanks, everyone. And so this is a great segue, and thank you, Jake, for all that into kind of the clinical experience with these drugs, including the clinical trials and kind of a little bit of our implementation of early COVID therapeutics. So, first and foremost, so this is a typical case, fairly similar to what we've been seeing recently. The vast majority of people who are vaccinated who get COVID will do well, especially if they are immunocompetent and can make a reasonable response to the vaccines. But there are, the needs for patients who, for reasons being higher risk or being immunocompromised, do need access to these molecules for the sake of making sure that they have a benign recovery as well. And so this is a typical case, a 65-year-old with cough, malaise, and congestion for 48 hours. His son recently contracted COVID from hockey practice. He has diffuse large B-cell lymphoma, is on rituximab, which is an anti-CD20 monoclonal antibody, which essentially leads to prolonged blockade of CD20 response, as well as CHOP, so cytotoxic chemotherapy, has diabetes, hypertension, and prior NSTEMIs. He's received 3 doses of an mRNA vaccine, although his antibody titers show no response. And he managed to get a COVID test that's positive and calls his oncologist to discuss his therapeutic options, giving his high-risk status. So, again, this is someone where on paper, despite them being fully vaccinated, you have significant concerns about A, their response to a vaccine, and B, their underlying medical conditions, which may predispose them to severe outcomes from COVID-19. And so what's approved right now really has 2 pathways. One is modifying the host response, we for early therapeutics, and Jake mentioned a number of kind of immunosuppressives, the ones that have been looked at in outpatient, particularly that have had clinical data. Fluvoxamine, which again was mentioned as part of the Together platform, which was McMaster led, as well as inhaled corticosteroids, which was led through Principal, Stoic, and a number of other clinical trial groups. I'm not necessarily going to talk about those today for the sake of really describing kind of direct antiviral agents or antiviral compounds, but certainly they play a role and are now recommended by the Ontario Science Table Guidelines for the treatment of COVID-19 in high-risk patients. But there is a number of pathways now to target the virus, which is very different than 2 years ago. So we have Mullen Ipravir, we have Paxlovid or Navier, which is the other name, Remdesivir, which is an old drug that we used in the hospital that's now may have benefit to be repurposed in the community, and monoclonal antibodies, which I'll mention. And we'll go a little bit over about implementation and our experience with monoclonal antibodies. So first and foremost, these are not therapies for everyone, right? So a 20-year-old who is fully vaccinated, who has a very low risk of a breakthrough infection leading to hospitalization, who is at baseline at low risk of a hospitalization, probably is not the person that you want to be offering these therapies to, because the number needed to treat to change the outcome of a breakthrough infection probably goes into hundreds or thousands range. If there was unlimited supply, then absolutely. But again, we probably do need to think about therapeutics in the background of patients that we know are going to be overrepresented in hospital, recognizing that the number needed to treat in those contexts is going to be significant. So, we talk about conditions like moderate to severe asthma, cancer, diabetes, intellectual disability, and congenital syndromes, cerebrovascular and cardiovascular disease, obviously states of immunosuppression like immunocompromised, HIV, solid and blood stem cell transplant, and cancer, and obesity, which is definitely a risk factor that's under recognized for severe outcomes. And so, again, when we're targeting these therapeutics, and you'll see the theme through much of the clinical trials, is to incorporate these people as the people that were offered therapy as compared to younger, healthier individuals, where you may not be able to show as much of a therapeutic effect. So I'll talk about Molnupiravir. This is a cytosine inhibitor, a cytosine analog that essentially causes chain termination as it's incorporated in RNA synthesis. And this was a trial looking at 1-to-1 different doses versus placebo. And again, when you start looking at groups that are increased risk, you start seeing the effects of these drugs show through. So overall, they showed very little benefit, 5.4 versus 3.1% of hospitalization and death. But as you start stratifying out for people at increased risk, and shorter symptom onset, which is the other part, is, as we know, antiviral compounds work very early. They reduce cytopathic effects and direct viral effects if modulated early. But as the disease progresses, it becomes as much an inflammatory disease where drugs like dexamethasone may help, as compared to an infectious disease where again, direct acting antivirals may help. So you see, again, if you get to earlier treatment and if you get to participants at higher risk, you start seeing the effects of the drug really take place. And this was the phase 2 data of the MOVE-OUT trial. The MOVE-OUT trial was finally published actually a few days ago. It's a bit of a weird story of how this is. There's a lot of anticipation of this. So they had done a large phase 2 trial and phase 3 trial. Many of the centers are international. And the one thing I will point out is this risk of hospitalization, which was about 14% in the placebo group, which is actually quite different than any other clinical trials of early therapeutic options that are mentioned. Usually we're seeing the hospitalization risk in a high risk cohort being between 7 and 10%. So 14% is quite high. Merck released their initial release of the first 700 people per group, and saw a 14% hospitalization risk in placebo and a 7% in intervention. Again, even in the intervention group, this is actually quite a high hospitalization risk. So you're picking very sick patients or patients that may be very close to hospitalization at the time of administration. the IMB actually stopped the trial to potential benefits and told Merck to go to the FDA and health organizations for approval. And interestingly, when they do a second release, by the time the IMB, the Data Safety Monitoring Committee had stopped the trial, there were another 600 or so enrolled in each group. When you now added those 1,300 people together in each group, you actually saw a much lower magnitude of benefit. So it went from 14 versus 7% to about 9 versus 6% after full analysis, with a 30% relative risk reduction and a 3% absolute risk reduction, number needed to treat of about 33. It is interesting when you actually look at that 600 group that were in as part of the release. They actually didn't really show much benefit to the drug, and so it's really that consolidated early treatment benefit group versus this benefit group. And there's a lot of questions at why that happened. Was it particular sites that were higher risk? Was it study protocols? And this is what led to, as Jake had mentioned, the really tight decision in the FDA for approval of this drug, recognizing that there are some inconsistencies between that first 700 and 600 patients that were enrolled in the trial. It's likely of some benefit of smaller magnitude. This is the Kaplan-Meier curve from the New England Journal publication, which does really show a time gradiented effect. But again, there are some really inconsistent issues that do need to be explained as part of this, and that have not been reconciled. And so, again, this may be a group where, if they are contraindicated to other options, may be considered for this, but, there is some question about how this compares to other direct antiviral therapeutics. So Paxlovid or Navier, which Jake had mentioned, which is complex with ritonavir, which is mainly used as a booster rather than necessarily its protease effects. This is a large multi-center trial, and again, the first 1,200 patients were published. The second update actually was consistent. So, given within 3 days, a 0.8 versus 7% risk of hospitalization. Within 5 days, 1 versus 6.7. There were only deaths seen in the placebo arm. It was well tolerated with minimal drug and effects. Certainly, there are patients, and these are protease inhibitors. They inhibit a major pathway as part of drug synthesis. So older patients who are on anticoagulants, steroids, anti-seizure medications do need to be monitored while they're on these, knowing that you may increase the dose of those medications and lead to adverse effects. And again, this one, contrary to what happened with Mullen Ipravir, the repeated data review after the IMB closure of the trial actually showed a more consistent effect, and actually 3 versus 5 days being fairly similar in terms of its effects. And so, there is a lot of hope behind this molecule, despite, again, the long supply chain issues, which Jake had mentioned, that this may be the one to help with patients early, and obviously with the benefits of being an oral therapy as compared to intravenous with the other therapeutic options I'll mention. Remdesivir is a direct RNA polymerase inhibitor. There was an initial trial at the beginning of COVID between kind of April and May looking at this in hospitalized patients. 2 trials, actually. One, the ACT trial, which was done in the United States, and the other, the Solidarity trial, which was the multinational trial done by the World Health Organization, including throughout Canada. And so, in the inpatient standpoint, there was a controversial benefit that was seen in early oxygenated patients to prevent them from progressing to ICU or needing ventilation, as well as being discharged early. There may have been a mortality benefit in that group, but as patients got more progressive into their disease, needing ventilation, there was no benefit seen. And this was confirmed both in ACT and in Solidarity. But there's always been a question, when patients hit hospital, it's been 5 to 7 days usually since their symptom onset. And so, again, are we getting into a point where the disease is much more inflammatory, where an antiviral compound may not offer as many benefits, and that low oxygen group may just be a surrogate of very early individuals? And so what would be the benefit of giving this drug very early? And so this was a randomized clinical trial that was done of patients getting 3 days, a single dose IV on day 1 of 200 milligrams, and 2 doses of 100 milligrams subsequently. and actually saw remarkable benefits, an actual 80% relative risk reduction of COVID-19 medically attended visits or death, which was seen, in these Kaplan-Meier curves, and comparable to some of the monoclonal antibodies and Navier in terms of its efficacy. There are obviously concerns of bringing someone with COVID-19 in for therapeutics for 3 days. But, again, a compound which I think we may have been misusing in the hospitalized setting, not to say it doesn't work there, where the benefits may actually be much more targeted towards the community and higher risk patients. And these were patients over the age of 60 or patients with one major comorbidity. When we talk about monoclonal antibodies, there are 4 that have been looked at: BAM, etesevimab, casirivimab, imdevimab, sotrovimab, and AZD7442, which I think was given a better name, which I can't remember right now. These are slightly different. So some are anti-spike, some are conserved elements, some are ACE2 receptor interfaces. There are changes with the FC region for longer half-lives. Sotrovimab and the AstraZeneca compound AZD are longer-acting monoclonal longer MABs for FC binding changes and different epitopes. So I'll go through them quickly. BAM etesevimab, this was a phase 3 trial done in high-risk patients. They saw a 70% relative risk reduction in hospitalization amongst patients who got a single dose. So symptom resolution was shorter, and certainly that has been our observation with all of these, that people feel better when they get these drugs. Regencov, which is the famous Trump drug Regeneron. So this is casirivimab and imdevimab, a large clinical trial by Weinrich et al. In the New England Journal of Medicine. And again, this was a trial that changed a little bit throughout it as dosing was reconsidered. But again, a trial that was stopped due to its effects from the Independent Data Safety and Monitoring Committee, where they saw again a reduction from about 3 to 4% hospitalization to about 1% hospitalization amongst people who got the drug. Relatively safe. There were actually more adverse events in the placebo group. No anaphylaxis or adverse events in that grade. And again, events were pretty similar for both groups, but certainly again a big shift in relative risk. And this was the largest monoclonal antibody distributed throughout the world, and really was taken up very aggressively in the United States and eventually in Canada once it was approved. Sotrovimab was also trialed in this trial. Actually, the initial site was done here in Canada at William Osler. And so looked at patients at higher risk, symptom onset less than 5 days. And similarly, as compared to Blaze and the Weinrich study, showed a marked relative risk reduction in hospitalization from about 6 to 7% to 1% amongst individuals. No one required a mechanical ventilator, mild adverse events, and again, more in the placebo group as compared to the hospitalized group. Sorry, in terms of the intervention group. And then Omicron came along and really changed our market to say, hey, we may have all of these really active compounds to Delta. So again, the Lilly compound for BAM etesevimab, Regen, the 2 antibodies, the Lilly drug is etesevimab, which is the one that's still active. The Regen compound, the AstraZeneca compound, and Vir, which is sotrovimab, which looked like they all had really good IC50s to Delta. And so we were very happy when Delta was here because we had access to these compounds. And then Omicron came with its many spike mutations and basically destroyed many of our monoclonal antibodies. It's still not clear as that the AZ compound has had a number of different studies which are showing IC50 in live virus versus pseudovirus assays being a little bit different. But the only compound which is still active, that is on the commercial market still, is Vir 7831, which is sotrovimab. And so many centers, including the United States, which had really plentiful supplies of Regeneron, lost their supplies essentially overnight and were forced to pivot to very limited supplies of sotrovimab, which in Canada have been limited as well. DXP 604 is a monoclonal that is used predominantly in China that hasn't made it to the commercial market yet. And so we were one of the first sites in Canada to start a monoclonal antibody clinic. Our model was 7 day a week. We do 5 days and weekends for urgents or high risk patients. There's beds actually, or chairs for 6 infusions at a time now. There's 2 HEPA filters in the room to reduce aerosol risk. There's a TV with CP24 on, so people have things to do. We've done up to 10 infusions a day. They're about a half hour to an hour apart, with an hour of downtime for people to watch for adverse reactions as part of the Health Canada spread, and separate entrances with exits for escorts. I'm going to present our data for the first data analysis, which was actually predominantly Delta. We are just starting to look at our Omicron data, but have been so busy that we haven't been able to consolidate it. Our first data kind of at the emergence of Omicron, and when things were predominantly Delta, were the majority of patients who got the therapy were unvaccinated individuals over the age of 50. There were a few under the age of 50 who were unvaccinated, who were high risk in immune compromised conditions. And again, there have been partnerships across the region and really across the GTHA to make sure that patients have access to this through our center and other centers. So we've been giving escalating doses per week, so 2 to 4 on a typical day. Again, this is up to week 49, week 51 and 52, and now week 1 of 2021. We've been giving about 40 doses a week, so we've certainly accelerated at this point. Most people were in within a day or 2 of their test, and again, this is when testing was still reasonable, and again, a good number of patients that were coming in towards the tail end. The colors didn't come out here very well, but the vast majority of people were over the age of 50 and unvaccinated. There were some that were unvaccinated and 50, and then there were some that were vaccinated and immune compromised. Majority of transplants and people who are on TNF alpha inhibitors or other therapeutic monoclonal antibodies for underlying inflammatory syndromes. Overall, and again, with the limitations of patient confidentiality, there was well tolerated, no adverse reactions. There were very, very few patients needing admission, and the vast majority of patients that needed admission on the day of their infusion. So they were not patients that were theoretically incorporated in the clinical trial. They should have been patients that were in hospital. There's been no deaths or patients ventilated that we've given a monoclonal antibody to. And again, in calling a lot of these patients, they had an immediate benefit within 2 to 3 days of the infusion. They actually, it's interesting, they had kind of a Yerix-Herxheimer reaction or kind of immune reconstitution reaction the night of the infusion. So people felt a bit lousier and felt like they just needed to go to bed afterwards, but woke up the next morning feeling better. And then the next day after that, feeling even better. And that's really been the pattern for many of them. There are barriers. And so, again, not all patients are eligible by the time they're identified. And certainly our testing issues recently have really led to this being a problem. There is hesitancy amongst unvaccinated people to get a therapeutic intervention. We've done a huge job, and the nurses in the clinic, who are incredible, who have really been champions now to run this as a almost autonomous clinic, have been great in being completely unbiased and completely encouraging of patients, and really, really lovely in terms of their experience. Transportation is an issue, and again, people have to drive themselves or get people to drive themselves to the clinic, which means that either they have to drive themselves feeling crappy, or they have to have positive household members that can drive themselves. If they're in a household that's isolating, where one person hasn't had COVID and one has, again, it's really hard to, it's not ethical to expect people to expose themselves to COVID to drive their family member to the clinic in order to access benefits. And certainly, when we were starting in October, there was a lack of awareness of early therapeutics, and in the United States has been taken quite aggressively. In Canada, patients didn't realize they had access to these drugs, and really, were hesitant to say it, as the previous paradigm was go home and rest, and if you get sick, come to the hospital. And so, again, as we work through the process moving forward with drugs like Nirmatrelvir, with more monoclonals. I think there are three major buckets here that really need to be dealt with. One is testing and aggressive early testing, especially to patients that are at higher risk. And we are now using that as part of our provincial pathway for patients to get tested, that they're put at the front of the line as we can intervene in that sense. So it's early, it's accessible testing. People don't feel stigmatized to go to testing. And that test result comes back to someone in a time-dependent manner where that 5 to 7 day window can be given to the drug for administration. You need assessment. And so you need people that are able to take tests and interpret them in the right patient. And so, people at the ends of assessment and testing centers that can do these assessments of appropriateness for things like Navier, Ritonavir. There are a lot of drug-drug interactions, so particularly our elderly and frail patients. You really want to make sure someone, a pharmacist and a healthcare provider that's familiar with drug-drug interactions, can counsel patients through it, can recognize what drugs can be held and what drugs need to be continued. And you need delivery. So, again, you need IV delivery, which is not an easy process. You need PPE, you need a well-ventilated space, you need training, you need medical staff, which are, the hot commodity these days. And even with oral therapeutics, you need someone to deliver the oral therapeutics. And again, we certainly have lots of options in Canada with, delivery services and others, but you do need to build that into the system that the drug can get to people. They can't just wander into a pharmacy. It needs to be there and counseled through in that sense. And with that, I'm happy to take questions and happy that we got a good chance to go over what will be a nice therapeutic window going forward. I will say one thing is a nice little anecdote at the end of this. The last time I presented at IIDR was January of 2020. It was with Aaron J. Banerjee, who's now at Vito in Saskatchewan. Aaron J. was talking about his work on bats and coronaviruses, and we were talking about Ebola at the time, recognizing that there was an outbreak in Kivu in the DRC, and we were talking about therapeutics for this, as well as so I was talking about remdesivir as well as monoclonal antibodies to another disease. And I remember very, very clearly at the end, Aaron J. putting up a slide about this weird stuff that was happening in China, and all of us were kind of just shrugging and thinking about Ebola in the DRC as the next major issue. And now 2 years later, I'm talking about remdesivir for the outpatient treatment of COVID-19. So things have kind of come full circle from that, as well as monoclonal antibodies.
Thank you, Zain. It's been quite a ride for the last 24 months. Okay, so we have a few questions in the Q&A. Des commented that Gord Guyot's been talking about exaggerated effects in RCTs stopped early for benefit for some time.
Yeah, and I think the Mullen Ipravir example is perfect for this, right? Like this thought of IMBs stopping trials and then reorganizing, like, yeah, we want people to have access to therapeutics, but we also need to get these trials completed. Mullen Ipravir, no one knows what's going to happen with it on the market, and that's really one of the real pressures here. If they published their full clinical trial, we would have been a little bit more, scratching our head saying, is this actually worth it in that sense?
Mm-hmm. I personally, some of the stuff I read made me worry that its mode of action may lead to more mutations, which we certainly don't need. So Des also said, Zain, studies of previous monoclonals suggested there was no apparent benefit in those that were seropositive. Can you speak to why this appears not to be true for sotromevibib?
Yeah, so it's a good question, Des. So Recovery, which was the large trial that was done in inpatients of monoclonal antibodies, saw no benefit in people that were seropositive. It's interesting, though, there is some data that has emerged in the outpatient realm, which is not as clear. And patients and there was a modified score, I forget what it was called, but patients that had breakthrough infections with a high modified score, recognizing that breakthrough infections, are often associated with lower antibody titers in some individuals. There was a suspect that maybe these would work better in high risk breakthroughs. So not everyone, but high risk breakthroughs. And so, there was a little bit of data published by Regeneron on this and showing that patients with high comorbidity scores may still benefit, and the number needed to treat was not trivial. It was like less than 10. And so this is where I think the use in outpatients, it may have been much more aggressively pushed, not necessarily for sotrovimab, but just for outpatients in general. But it is a piece that we're still struggling with, and now with Omicron and potentially lower severity amongst vaccinated individuals, it's even harder to sort out what's the number needed to treat here and what are the core groups that need treatment in that sense.
Mm-hmm. Andrew MacArthur comments, Delta may not have been around long enough to develop resistance, but if Omicron stays in the community for a long stretch, what are the odds that a subvariant will undermine treatments?
Yeah, it's a good point. I mean, the companies are developing Omicron-based monoclonals. So Lilly and Roche are developing them. GSK has the sotrovimab compound. It's interesting. Sotrovimab is an element that was conserved from SARS-CoV-1, and so perhaps that may be an element that may not be as plastic over time and would be conserved over time. But absolutely, look, we're using these in immunocompromised patients. These are the patients that, if not followed correctly, may be the ones that harbor mutations that lead to variability. And we've seen this. There's lots of great Nature publications about prolonged COVID infection and just the accumulation of mutations amongst them. So absolutely, look, they're time limited, and if you're not clearing the virus out quickly, or people are using these drugs indiscriminately in high-risk patients, there is a risk that mutations develop to these therapeutics.
Mm-hmm. Yeah, I actually have a question for Jake related to that. So, it struck me when you showed that diagram of the genome of the virus and all of the polypeptides that it can make, that we're still very focused on a small number of targets, right? And presumably the proteases are a little bit less likely to mutate than the spike, but what are the chances that these protease inhibitors are going to drive the emergence of a resistant variant, and that's going to actually spill over into other viruses as well?
Well, one interesting aspect of that, Laurie, is that the active site on the main protease of SARS-CoV-2 has 100% sequence homology with the one from 2002 with SARS-CoV-1, right? Which I don't think I've ever seen. I haven't seen very much biology, but you don't often see 100% sequence homology. When I saw Dr. MacArthur's question, I thought, Andrew, you're probably the best person to answer your own question out of the people that I know. What's the likelihood of resistance? But my intuitive hunch is that the proteases are pretty immune to mutations. So I would think that if any of the drugs were to be longer lasting, and maybe even pan inhibitors against novel pandemics, it would be this Paxlovid out of Pfizer and other compounds kind of like that I'm sure we'll see in the coming years.
And you mentioned that the compound's very challenging to manufacture. Once they've identified a compound like that and they know how it works, is there a possibility to make something that's a little less complex or a little more amenable to bulk production?
The short answer, I think, Laurie, is no. That idea of let's make it as simple to make as possible, that's done preclinically, right? Once you go through human trials, you've committed to a molecule, that you must make it, right? So everyone's trying to generate the simplest possible, most synthetically tractable compounds. But there are some really cool synthetic technologies emerging right now, including evolved enzymes that modify molecules in ways that you would not expect, and compounds that are difficult to make become easy to make using these new technologies. So I think everything is scalable. We saw recently a talk from somebody at Merck about the Malu Navier synthesis. So the one they're using now to supply the world is 8 steps, but they're developing one that is just 3 enzymatic steps, and it's all done in bioenzymatic reactors. I'm not aware of Pfizer doing the same thing for this one, but It's just old school synthesis. I mean, it really is a lot. Like a 500 tons per year is a massive effort for a molecule of this complexity.
Amazing. And last question for Zain. If Health Canada approved these drugs tomorrow, what would you reach for? Would you reach for a monoclonal, or would you reach for the drug?
Yeah, I mean, Navier, I mean, look, oral therapy is going to be a whole lot easier to mass administer as compared to intravenous therapy, right? You can get more doses out in a given time span. So, I think if it were to get released tomorrow, I think the way we do assessment and testing, number one, may incorporate a little bit of assessment for therapy at assessment centers for high-risk patients using, rapid molecular and rapid testing to actually have access to a result for high-risk patients and tie them to getting drug right then and there. But yeah, I would, if I could scale up on something, it would be the protease inhibitor and use monoclonals for things like major immunocompromise where they may have more of a niche in that sense, given the ability to scale up with oral therapy a whole lot better than intravenous.
Okay. Well, great. Thank you both again for those fantastic talks. It's just truly amazing to see how when people row in the same direction, how much progress can be made in such a short time. It's impressive. So thank you everybody for joining us, and we'll see you next month for rounds on February 2nd.